Okay, let me see if I can find out how to do the do
Sharing.
this properly. Share screen, um, advanced portion of screen, share.
There you go.
Okay, so thank you for the introduction. I'm happy to participate in this series of presentations. Let's see. So I have received funding from Biotech Pharmacal in Fayetteville, Arkansas, a supplier of vitamin D and other supplements. As mentioned, I have a PhD in physics, and I have studied health since 1996, resulting in 324 publications on solar UVB and vitamin D, and 54 regarding diet. These publications included articles, reviews, editorials, and letters to the editor. So the outline for today is, first of all, vitamin D physiology, then types of studies used to find out how something like vitamin D affects health outcomes. Then the health outcomes I want to discuss include cancers, infectious diseases, pregnancy outcomes, diabetes mellitus, cardiovascular disease, dementia, and autoimmune diseases. Then, of course, there are some concerns about sunlight exposure and vitamin D supplementation. And at the end, I'll have recommendations for how to get the most out of vitamin D and sun exposure. So in terms of the vitamin D physiology, vitamin D3 is produced in the skin by solar UVB radiation. That's from 290 to 315 nanometers, hitting 7-dehydrocholesterol in the skin, followed by thermal reaction. So this UVB radiation represents, during the middle of the day, 3 to 5% of the total UV, which means it's much less than 1% of total solar radiation. So once the vitamin D is produced, it enters the blood, and in the liver, it receives a hydroxyl group to become 25-hydroxy vitamin D, or calcidiol. Now, this is the metabolite measured as for the vitamin D level when you have your blood drawn. But that's not an active metabolite. When it receives another hydroxyl in the kidneys or other organs, it becomes 1,25-dihydroxy vitamin D, or calcitriol. And that's the active hormone, active version of vitamin D, and the hormonal one. Turns out that as organs and cells need the calcitriol, they can convert the 25-hydroxy D to calcitriol, so that this helps maintain calcitriol levels at the proper level for helping to regulate serum calcium and other things. So here's just a schematic. You see in the upper left the vitamin D production and sources, goes to liver, and then the what's called a prohormone, the 25-hydroxy vitamin D, and in the yellow circle, you see the effects, the benefits of vitamin D in affecting disease, such as cancer and other things. And then in the lower left, you see the effect on bones and calcium absorption. Until about the year 2000, vitamin D was primarily thought of as very important for bones, reducing the risk of rickets, and maintaining proper calcium levels in the blood. But it wasn't until about 2000 when work really started to accelerate in finding all the other health benefits of vitamin D. And as a result, this is only 23 years or so after the big push on vitamin D, we're still learning about the effects of vitamin D, and they're not fully accepted by the medical system. So calcitriol exerts many biological activities by regulating transcriptional activation and or repression of target genes. In order to achieve this, calcitriol binds to the vitamin D receptor. That's a nuclear transcription factor that's present in almost all tissues. Thus, it is a hormone. Hormones can help control how cells and organs function. It was named a vitamin because when it was discovered around 1922, they had discovered vitamin A, vitamin C, vitamin E. Well, anyway, and they just thought that this was another simple hormone, but is now quite well understood that it's a hormone, but they retain the name vitamin D. Now, there are both genomic and non-genomic actions of vitamin D. So we just discussed the genomic effects. And the non-genomic ones are when the vitamin D receptor induces rapid signaling situated on the cell membrane and or cytoplasm. Emerging evidence supports the notion that vitamin D enhances immunity, providing protection towards pathogens, while concomitantly it exerts immunosuppressive effects by preventing the detrimental effects of prolonged inflammatory responses to the host. This is another schematic. On the left, you have the genetic regulation, and on the right, you have the non-genomic regulation. I've dealt mainly with the genetic effects, and that's what I'll be discussing primarily today. So here's a demonstration that vitamin D affects gene expression. This is an experiment reported in 2019. 30 healthy adults were randomized to receive 600, 4,000, or 10,000 international units of vitamin D3 for 6 months. A plateau in the parathyroid hormone levels was achieved at 16 weeks in the 4,000 and 10,000 IU per day groups. There was a dose-dependent 25-hydroxyvitamin D alteration in broad gene expression with 162, 320, and 1,289 genes up or down-regulated in their white blood cells, respectively. It is now known that vitamin D affects maybe 5% of the total genome of the body. So it's very important. It's also been shown that there's a large variation in gene expression between summer and winter. Now, that could be due to changes in vitamin D. It could also be due to photoperiod changes, length of the day. It could also be due to changes in temperature. Not clear, but anyway, it's important that vitamin D does do a lot of its work through affecting gene expression. So the sources of vitamin D. Primary source for many people is solar UVB, but you can only make vitamin D when the solar elevation angle is greater than 45 degrees. Now, there's a shadow rule. The dermatologists say if your shadow is shorter than you are, stay out of the sun or get sun protection. But you can't make any vitamin D if your shadow is longer than higher than you. So we have sort of the opposite shadow rule from the dermatologists in terms of making vitamin D. Now, in terms of foods, which may produce maybe 5 or 10% of vitamin D levels, fish, mostly cold water ocean fish. And the best ones are those low on the food chain, which would be cold water salmon, wild salmon, not farm salmon, sardines, anchovies, herring, mackerel. It turns out that meat is also a very important source of vitamin D. That's in the form of 25-hydroxyvitamin D. There was a study reported from England in 2011 that found that meat eaters had the highest 25-hydroxyvitamin D concentrations, followed by fish eaters, and much higher than vegetarians or vegans. There was an 8 nanogram per milliliter difference between the meat eaters and the vegans. So, so this means that depending on your diet, you may have to look for other sources of vitamin D. The fortified foods such as milk and flour products are also available in some countries, but do not supply very much vitamin D. So this means that if you're not spending much time in the sun in the middle of the day, especially in the summer, you've got to rely on supplements. And there are two types. There's vitamin D3, cholecalciferol, and vitamin D2, arylcalciferol. So vitamin D3 is the natural form of vitamin D, which is what we make in our skin. And that is the preferred type of vitamin D. And that's what most supplements are now that you buy in the vitamin shop or in the grocery store. Vitamin D2 is made in yeast or fungi, and it has been patented and can be sold through pharmacies. And so physicians often recommend vitamin D2 because they can go to the pharmacy and the patients don't have to pay for it. However, it's much more expensive than vitamin D3, and it's not as effective. So we recommend vitamin D3. Now, it turns out that the skin has all the mechanisms for converting the cholesterol to vitamin D3, from vitamin D3 to 25-hydroxyvitamin D, and from there to calcitriol. If you are in the sun, you will eventually make vitamin D to help protect your skin. But this is a study reported by dermatologists reporting that supplementing with enough vitamin D to raise the 25-hydroxyvitamin D to above 40 or 50 nanograms per milliliter, that's over 100 to 125 nanomoles per liter, would be able to attenuate the inflammation from sunburn. And a colleague of mine whose wife has red hair and freckles told me that when she gets her 25-hydroxyvitamin D up to around 70 nanograms per milliliter, she can go out in the sun in Illinois and spend an hour in the sun without any reddening, without any burning. So, of course, the dermatologists are recommending that you should use sunscreen, but the sunscreen has problems in that it primarily blocks the UVB, which produces vitamin D, and doesn't reduce the transmission of the longer wave called UVA as much. And it turns out that UVA is more responsible for melanoma than is UVB. So there's a problem with that. Also, the body's natural approach for reducing sunburn and cancer is to gradually tan throughout the season. If one goes out every day and spends a little time in the sun, as the spring and summer arrive, one slowly builds up a tan that reduces the penetration of UVB by a factor of 2 to 4. And that's generally enough to protect the skin while one is in the sun. Now, the dermatologists often point to the fact that melanoma is due to sun exposure. Well, if you look at the cancer statistics for the United States for 2018, you see about 1.7 million new cancer cases and over 600,000 new cancer deaths. The melanoma to cancer cases is only 5%. The melanoma to cancer deaths is 1.5%, and melanoma to all deaths is 0.35%. So as I will show, UVB protects against many cancers. So the net benefit is, you do a net benefit. You just have to be a little careful not to sunburn and overexpose. Now, here's a chart showing how long it takes to produce vitamin D as a function of latitude and season. So in the bottom, we go from January through December in the Northern Hemisphere. And on the left, we go from latitude from the equator up to 90 degrees. On the right, the color scale starts with 1 minute. Well, the black means it doesn't take much time at all. And the pink means it takes about a minute. You get up to the blue is taking maybe 12 minutes. So that's for 400 IUs of production with hands and face exposed for type 1 skin pigmentation. Now, turns out that the pigmentation for many people is much darker. Type 1 is very pale skin, like in Northern Europe. Type 5 is like in Africa. Type 3 might be Hispanic or Middle Eastern. People generally, people who've lived in an area for hundreds or thousands of years generally have the skin pigmentations that's ideally suited for where they're living. Now, take Australia. The Aborigines who've been there for thousands of years have very dark skin. But as you can recall, many of the inhabitants of Australia were brought there as prisoners from the United Kingdom. And they have the Celtic skin, which is very fair, freckled, red hair, and so on. And they have a different type of melanin than most people do. And their melanin, I think called pheomelanin, does not tan very easily. And as a result, they are very sensitive to the skin. And if they spend much time in the sun, they can easily burn. And so for years, the Cancer Society in Australia has been very cautious about recommending sun exposure to Australians. And it got to the point where up until a few years ago, they had more or less convinced the Australian vitamin D researchers not to promote vitamin D for fear that they would let the Australians spend more time in the sun and develop more skin cancer, melanoma. They did outlaw indoor tanning in Australia several years ago, and I think it's also not very popular in the United States anymore. So, as I'll explain later, while the skin pigmentation can be ideally suited for people living where their ancestors lived, if they move to another location, like to Australia or to the United States, if they move to where their skin is much darker than the prevailing skin pigmentation, they suffer. They can become vitamin D deficient. Now, there is a seasonal variation in 25-hydroxy vitamin D concentrations. This is findings for 45-year-old inhabitants of the United Kingdom. On the left, we have these 25 going up to about 75 nanomoles per liter, which is about 30 nanograms per liter, and down to 35 nanomoles per liter, or about 15 nanograms per milliliter. There's 2.5 nanomoles per nanogram, if you want to remember the calculation, conversion factor. So the minimum concentration is in February or March. That's because vitamin D slowly decays as a function of time. And also the peak is in September because it still takes time to build up. It's just like the temperatures, the longest day of the year is in, say, June 21st, but the Earth is still warming up because it's still adding heat as time goes on. The same thing happens for vitamin D. We have the same effect in the United States. Paper from 2015 showing the results from 2007 to 2009. This time we have the 25-hydroxyvitamin D in nanograms per milliliter, going from about 20 to almost 30 as a function of season. So, so the question is, if we're not making vitamin D from the sun in the winter, how do we maintain vitamin D in the winter? Well, the answer is provided by our colleagues in Australia. They pointed out that skeletal muscle cells store vitamin D as 25-hydroxyvitamin D by incorporating the vitamin D binding protein. And when intracellular vitamin D binding protein undergoes proteolytic breakdown, the bound 25-hydroxyvitamin D is then released and diffuses back into the blood. Parathyroid hormone is the most likely factor enhancing the repeated cycling of 25-hydroxyvitamin D between skeletal muscle and blood. This mechanism seems to have appears to have evolved to maintain an adequate vitamin D status in winter. And as I mentioned, this is also why red meat is an important source of vitamin D, which was only realized, like in 2011, which means that a lot of the food frequency questionnaires, which are used to evaluate the amount of vitamin D in blood or in food, have not incorporated meat as a source of vitamin D, as so may underestimate the amount of vitamin D from food. Now, this is a graph from a chapter in the Feldman and Pike's Vitamin D 5th edition, edited by Martin Hewison et al. It's a big 2-volume book, and this is chapter 56. What I did here is I got all the data I could for 25-hydroxyvitamin D in summer and winter for European countries. Now, this is in 25 in nanomoles per liter. And you see that in summer, the average level from 35 degrees north latitude, which would be, say, Greece, to 65 degrees north latitude, which would be Iceland, is around 67 nanomoles per liter, or around around 28 nanograms per milliliter, which is surprising since in Northern Europe, they have paler skin. In Southern Europe, they have darker skin, et cetera, et cetera. But now in the winter, you have a curved, a U-shaped relationship that in Northern Europe, they have concentrations nearly the same as what they had in the winter, summer. There's the most variation around 50 degrees north latitude, and at around 35 degrees latitude, they start to have higher values compared to mid-latitudes. So to explain this, in the summer, there are many effects that modulate the 25-hydroxyvitamin D concentrations. For example, in low latitudes, it's hotter, so people don't spend as much time in the summer and when it's really hot. That also in the mid-latitudes, lower latitudes, they tan, which means as the season progresses, they're making less vitamin D for each time they spend in the sun. And as I'll explain, it's also that UV exposure can also reduce vitamin D concentrations as well as increase them. So if you start getting too much vitamin D, the UV exposure starts reducing the amount of 25-hydroxyvitamin D. So in Northern Europe, it turns out if you look at diets in the world, in the tropics, diets are much more likely to be plant-based, whereas in high latitudes near the polar regions, they're much more likely to be animal-based. Part of the reason is in the tropics, you can grow vegetables and grains and so on, and go out every day and find some food to eat. In the northern latitudes, you have several months of the year when it used to be iced in, snowed in, people had to live on what they had stored. or on animals like milk and meat and fish and so on. So since animal sources are the only source of vitamin D, the northern latitude people have more vitamin D from their diet than do southern latitude people, which helps explain the winter event in northern European. So here's how sunlight regulates cutaneous production of vitamin D by causing its photodegradation. So once vitamin D is formed in the skin, exposure to sunlight results in rapid photodegradation to a variety of photoproducts, including 5,6 trans vitamin D3, suprasterol 1, and suprasterol 2. This was shown in a study by Webb and Hollick in Boston. Now, it would be nice if we could tell people that if you measure your vitamin D level and find out you have vitamin D deficiency, and we know your weight, your mass, your body mass index, and maybe we could tell you how much vitamin D you needed to supplement in order to raise your vitamin D level to where you want to be. Unfortunately, as has been shown now very well by Christian Karlberg and colleagues from Finland, now in Poland, that there's what's called a vitamin D response index. So when vitamin D, recall that you have a large chain of events going from the 7-dehydrocholesterol to calcitriol. You have all these steps, and all these steps involve genes. Now, genes, as you may know, have different forms called alleles. So you may have 4 or 5 alleles for each gene involved in or more involved in these processes. So some of the alleles are more effective in converting vitamin D along the way to the other products, and some are less effective. So what they did here was they took 25 participants in the winter, measured their 25-hydroxy vitamin D, gave them a certain dose of vitamin D, and then measured their relative change in 25-hydroxy vitamin D. Now, the score, the scale on the left is from 0 to 100, but actually they saw about a plus or minus 20% variation in vitamin D level. So this is very significant, and this means if you're going to want to raise your vitamin D level by supplementing, you should measure your 25-hydroxy vitamin D before you supplement, and then measure it again after you've supplemented for 2 to 3 months, the time it takes for vitamin D to come to equilibrium. And then you can see whether you've made the right decision. Okay, now I'm going to get into types of studies because it takes a lot of work to show whether and how strong the evidence is that vitamin D affects health outcomes. So there are a whole bunch of studies. There's ecological studies, both geographical and temporal. These are often the first studies to find something. For example, rickets in the old days, I guess has more of an observational study. Observational study, you do case control studies where you compare those who have a disease to those who don't have a disease, what the difference is in their intake or vitamin D levels. Prospective studies where you enroll people in a study, you measure values of various factors at the outset, you follow them and see who develops different diseases. Cross-sectional studies, you measure parameters and health conditions of people. Then you have the interventional studies, the randomized controlled trials. You take people, you then randomize them to some receive like vitamin D and some do not. You compare the outcomes. You have observational interventional studies where you have an open label study. They all know what they're taking. They all know what their vitamin D level is. And you look at the health outcomes. and you can have animal studies. And then there are mechanisms where you're looking at, well, what happens to the cells and the organs with vitamin D. And then finally, a new kid on the block is Mendelian randomization or genetic studies, where they look at, they take a large number of people, look at their the alleles of the vitamin D genes, and find out whether they have developed the disease or not. Okay, ecological studies, the geographical ones use population defined geographically, and they compare outcomes, health outcomes with modifying factors, all averaged for the population level. And that's where I got my start in doing health research, and I'll show a number of examples of this. The temporal ecological studies investigate trends or seasonal variations of health outcomes with respect to various factors, such as either summer and winter development of infectious diseases. Here is an ecological study for dental caries or dental condition with respect to UVB. So these were men entering the armed forces, their dental condition was measured, and they assigned a rank. The higher the rank, the poorer the condition. And what I showed when I looked at plotted them versus the UVB in July for the state was that when you went to the very lowest levels in the Northeast United States, you had very high dental rank, very poor dental condition. When you got to where you had 7 or higher kilojoules per meter squared, you had good dental condition. And it's known that UVB, through producing vitamin D, puts more what are called cathelicidins and defensins in the saliva, which kill the bacteria that cause dental caries. They didn't understand that at the time, but they did show that UVB and vitamin D reduce the risk of dental caries. Observational studies, case control. I think I've already discussed this. The best known interventions studies are randomized controlled trials. Most random vitamin D randomized RCTs are based on the guidelines for pharmaceutical drugs. In drug RCTs, the only source of the drug is in the trial. Excuse me. And the control drug group does not receive any of the drug. In most vitamin D RCTs, participants have above average serum 25-hydroxyvitamin D concentrations, are given relatively low vitamin D amounts, and all participants are permitted to obtain additional vitamin D from the sun, diet, and supplements. In addition, results are analyzed according to treatment, not hydroxyvitamin D. As a result, most vitamin D RCTs do not find beneficial effects of vitamin D. So one wonders, are these done because of lack of knowledge and stupidity, or are they done this way out of intent to show that vitamin D doesn't work? Ponder that. It is possible, however, to do it based on serum 25-hydroxyvitamin D concentrations. What you have to do then is measure 25-hydroxyvitamin D before you enroll people. Try to enroll people with low vitamin D levels. Give them a vitamin D dose that's going to raise their level to high values that are going to reduce the risk of disease, and then measure their achieve 25-hydroxyvitamin D levels. These are just starting to be done this way. Mechanisms, as I said, remember, most of the vitamin D effects are through the genomic actions, through calcitriol binding to vitamin D receptors coupled to chromosomes. The human, animal, and cellular studies used to identify and study mechanisms. Knowing the mechanism strengthens the findings from population studies or epidemiological studies. As I mentioned, these are from genes. The best MR studies often include data from more than 100,000 participants. So these are calculation intensity. They are often use data from a large data bank like the UK Biobank. So physicians are taught to respect studies only from randomized controlled trials. Since most vitamin D RCTs have not been properly designed, conducted, or analyzed, and have not shown results of vitamin D that are beneficial, most physicians do not think that vitamin D has much benefit. Findings from observational and mechanism studies are considered by experts when they make recommendations regarding vitamin D. I published a paper a couple of years ago comparing evidence from observational studies and randomized controlled trials for non-skeletal health effects. And it's almost a difference between night and day. So now let's get into the various diseases. I cut my teeth on cancer and vitamin D. So the ecological studies, observational, RCTs, and the mechanisms. Here is some of the data I used in a paper I published in 2002. So when the Atlas of Cancer Mortality Rate was published in 1999 by the National Institute of Cancer, they showed mortality rates by state economic areas for about 30 types of cancer. And what you see here is they have a color scale from high values are red, mortality rates are red, low values are in blue. For colorectal cancer, it went from about 10 in blue to almost 30 in the red. And you notice that the red is in the northeastern quadrant of the United States, blue is in the south and the west. Now, on the left, you see solar UVB for July 1992. This is from the NASA satellite instrument called Total Ozone Mapping Spectrometer. It's the cloud-free distribution of solar UVB at the surface. It turns out that they called it DNA spectral exposure because the UVB also affects can affect DNA. I don't think it corrects for aerosols either. So there are three things that affect this and make it asymmetrical. In the western half of the United States, the surface elevation is generally much higher because of all the mountains and just the high desert and so on. So with less atmosphere to penetrate, there's more UVB, less scattering by the atmosphere. Then as the prevailing winds are from the west, and so the air mass has to cross the Rocky Mountains. And in doing so, it pushes up the tropopause and compresses the stratosphere. So we have a thinner stratosphere, and it's a stratospheric ozone that reduces the UVB coming through the Earth's surface. And then you have a latitudinal effect because there's more UVB near the equator than higher latitudes. Now, in the eastern United States, it's all pretty much flat land, and so then you have just the latitudinal effect. So this asymmetry really allows you to say whether it's UVA or UVB. If you go to Australia, you have the latitudinal effect, but you can't distinguish between UVA and UVB. So it turns out, so it was Cedric and Frank Garland who first identified the role of UVB in reducing the risk of colon cancer. They had maps from 1974 from the National Cancer Institute, which had only 5 colors, and colon cancer was the only cancer that had a really large variation. And see, they grew up in San Diego and had just driven across country to Annapolis to enroll in Johns Hopkins School of Public Health. And when they saw this in their first week of studies, and they recalled that it's very sunny in the southwest, and not very sunny in northeast. And so they realized it was sun exposure. They knew that the most important physiological effect of UV exposure is vitamin D production. So they said, aha, it's got to be that vitamin D reduced risk of colon cancer. It took them 6 years to get their findings published. And then it was in the British journal, International Journal of Epidemiology. The mainstream American journals did not want to publish this. It was just, did not believe it, did not understand it. So they went on to show that ovarian cancer and breast cancer were also linked to low UVB, but didn't get much traction. So when this was published in 1999, and I worked at NASA Langley Research Center at the time, I knew about getting the NASA data, and so I could compare the solar UVB doses for each one of these state economic areas for every cancer. And I spent a couple of years doing that. Here's the breast cancer map. And you see this is pretty much similar to one for colon cancer, except now along the West Coast, you have a little bit more cancer, especially along the coast. Well, it turns out along the coast, you have a lot of clouds, especially in the Bay Area. Mark Twain famously said that the coldest winter he ever spent was a summer in San Francisco, because the Central Valley brings in the marine air through the Golden Gate, and we have fog and clouds all summer. So breast cancer may be a little bit less sensitive to UVB and vitamin D than is colon cancer. Now, melanoma, yes, is stronger in the south than in the north, but it's not so much related to UVB. As I said, it's more related to UVA. But the highest mortality rate here is around 4 per 100,000 per year. And you go back here for breast cancer, it's 30 per 100,000 per year. So it's a small amount. Now, my ecological study was for Caucasian Americans from two periods, 1950 to 69, 1970, 1994. And the first study I did in 2002 was state average. The second one was state economic area average. The second one was state average in order to get the other confounding factors such as alcohol consumption, Hispanic heritage, lung cancer, poverty, and degree of urbanization. And it turns out that the effect of UVB was not changed by adding all these other factors. They just sort of perturbations along the general finding from UVB. But while the first study stimulated much additional research, the critics were not going to accept it without showing that the confounding factors had been accounted for. So it turns out that 15 types of cancer were found inversely associated with UVB. There were also 9 associated with alcohol consumption, 6 with Hispanic heritage, 10 with smoking, 7 with urban residents, and 8 with poverty. It turns out that smoking is also an index for diet, which I didn't know at the time. Now, however, it turns out that we have many different factors affecting cancer risk. So in the earlier periods, people in the U.S. spent a fair amount of time in the sun, but they gradually increased sun protection and reduced sun exposure in the second period. They were not obese, and cancer treatment was not advanced, so cancer mortality rates correlated well with cancer incidence rates. Thus, the UVB vitamin D effect was really apparent in the cancer mortality rate data. More recently, Americans have lower unprotected sun exposure. They wear sunscreen. They often stay out of doors. They're staying indoors, working on the internet, etc. They're more likely to be obese and to have diabetes, all of which affect cancer rates. So here is the new maps for this is the rate of new cancer in the United States, 2016 to 2020. This is colon and rectum combined, all ages for whites, non-Hispanics, males. And you see now, you still have higher rates in the north, but not in the north, so much the northeast. You have the strongest rates in the south, Texas, Louisiana, Georgia. This is due to the diet, obesity, smoking, and probably air pollution. And I'm working on a manuscript to explain this in more detail. Here's female breast. You see here that you still have some effect in the Northeast, but you have a lot also Northwest and in Texas. I think mammography is much more common now. So women are tested every year or two. And of course, obesity plays a role. On the other hand, corpus and uterus cancer still shows a strong UVB vitamin D relationship. Now let's turn to observational studies based on vitamin D supplementation. This is a paper from 2018. It's analyzing results for women who were either in one of two randomized controlled trials conducted by Joan Lappy et al. at University in Nebraska, Creighton University in Nebraska, or it was related to women enrolled as volunteers in the grassroots health volunteer cohort who would take whatever vitamin D dose they wanted to and have vitamin D levels measured every 6 months with a mail-in blood spot test. And so what you see here is that on the horizontal, we have concentrations out to about 80 nanograms per milliliter. And we have the frequency of cancer according to the fraction of participants who develop breast cancer in that the different concentration range. And you see it's almost a straight line decrease from one place to the other, from low to high. And so it's shown here that there's an 82% lower rate for those who had greater than 60 nanograms per milliliter compared to less than 20 nanograms per milliliter. Now, there are other factors involved. For example, those with the higher concentrations had a lower BMI, more like 24, 25, than those in the Nebraska studies who had a BMI more like 30. So BMI does also affects concentration. But you want to look here at the, say, going from 30 to 40 nanograms in the middle. It turns out that if you use only 2,000, the Nebraska study used 1,000 IU per day plus calcium. The VITAL study I'll discuss later used 2,000 IU per day. And so if you're going to use only 1,000 or 2,000 IU per day, you're only going to move from maybe 30 to 40 nanograms per milliliter. So if you measure just on the basis of were they treated with vitamin D, you're only going to get the effect of 10 nanograms per milliliter, which is not going to show very much. You really got to look at a large range of 25-hydroxyvitamin D to find a good effect on cancer. Now, it turns out another consideration in observational studies is that the longer the follow-up time the lower um, the observed effect. In other words, what I'm trying to say is that, well, 25-hydroxyvitamin D changes as a function of time. So does diet. So if you enroll people, say 12, and then follow for 12 years, you're not going to find much of an effect of vitamin D. And so what I've done is I've plotted the observed effect for high versus low 25-hydroxyvitamin D for men and for women, men in the dots, women in the circles. And I show that if you extrapolate to 0 follow-up, you'd find about a 23, 24%, 25% reduction in colorectal cancer. So when the paper was published, using the non-thinking way of put the data in and turn the crank, they did not find an effect of vitamin D, or found a very small effect. But this shows that there was a large effect if you consider follow-up time. So now we turn to the VITAL study. This was run by Joanne Manson at Harvard University. It enrolled over 25,000 participants, including over 5,000 African Americans. Half received 2,000 IU a day vitamin D3, and half a placebo for 5.3 years. There is also an omega-3 fatty acid study, which was different from this. So we say that half of these who received vitamin D also received omega-3 and half did not, but that did not affect the vitamin D study. The mean baseline 25-hydroxyvitamin D for those providing vitamin values who were treated with vitamin D is around 31 nanograms per milliliter. So that's even above the average 25-hydroxyvitamin D in summer for the United States. And all participants were permitted to take up to 600 or 800 IU per day, as well as spend time in the sun and eat whatever they wanted to. So you did not have a good control group. It turns out that those who did not take vitamin D, those who took vitamin D had about a 12 nanogram per milliliter increase in vitamin D. So what did they find? For the entire group, there was about a 25% reduction in all cancer mortality rate, but no significant effect on cancer incidence. Well, it turns out that vitamin D is more effective in reducing cancer mortality rate than it is in reducing cancer incidence. The simple way of thinking about that is that there are many factors that result in cancer. You have the effects of DNA damage by smoking, by alcohol, by pesticides, et cetera, et cetera. But when it comes to progression of cancer, you've got to have blood vessels grow around the tumor in order to bring nutrients to the tumor. And vitamin D reduces angiogenesis around tumors. It's one of the few things that does that. And then vitamin D also reduces metastasis into the surrounding tissues. Again, one of the few things that does that. So it's not surprising that mortality rate was affected stronger than incidence. Now, on the other hand, for those with body mass index less than 25 kilograms per meter squared, there was a significant 25% reduction in cancer incidence, but not for those with 27 higher than 25. For African Americans, there was a barely non-significant 25% reduction in cancer incidence. And that's because African Americans had lower mean 25-hydroxyvitamin D than the white Americans. Now, the abstract, which is what most physicians read, did not mention these beneficial effects. The study was published in the New England Journal of Medicine, and that's primarily an advertising tool of big pharma, and they have the rule that there can be only one outcome per study. And so they did not want to let the physicians know that there were some other benefits. If you looked under the hood, you would find them, but most physicians don't have time to look under the hood. And in all the press conferences, Joanne Manson didn't bother to mention these other benefits. Now, here's what they showed in terms of BMI, a 24% reduction for less than 25, no reduction for higher values. Again, it was about a 12 nanogram per milliliter increase in 25-hydroxybutyrate. Now, here's a very interesting study that just came out about a year ago. So the P53 gene is a tumor suppressor gene, and its activity stops the formation of tumors. They did a study where they were looking at the patients with digestive tract cancers, and they did a 5-year study on relapse or death. And they looked at a subgroup of patients who were P53 immunoreactive. That's defined as positivity for anti-P53 antibodies in serum or nuclear accumulation of P53 oncosuppressor protein in more than 99% of the cancer cells. So some of these patients had lost the use of their P53 gene. And the results are, if you look at on the left, you see that for those with the, these is those without the P53 gene working, those given the placebo almost all died, or we call that, with the placebo, they have very poor outcomes. Those with vitamin D had only about a 20% relapse. And if you look at those without the P3 effect on the right, vitamin D didn't have any effect. So this is about finding about a 16% reduction in mortality rate due to vitamin D supplementation and having the P53 effect being deleted, not working. So this is done for digestive tract cancers, but as far as I can tell, this would affect all types of cancer. And so this means if you get cancer, you probably want to have your oncologist find out, are you P53 immunoreactive or not? And if you don't have your P53 working, be sure that you supplement with high-dose vitamin D. The mechanisms, I've already discussed this. Well, the first thing is that vitamin D reduces cancer incidence by mechanisms such as surveilling cells and allowing good ones to differentiate and proliferate and build the organ, but causes cancer-forming cells to disintegrate through a process called apoptosis. Now, moving on to infectious diseases, the primary mechanisms, one is induction of innate immunity through secretion of defenses and human cathelicidin LL37, which have antimicrobial and anti-endotoxin activities. So innate immunity means that it doesn't matter what the organism is, virus or bacteria, it will attack them all. Now, there are also innate and also non-innate mechanisms that are related to particular organisms. In other words, the body learns to react to perhaps influenza or tuberculosis or something. But now vitamin D also regulates the chemokine and cytokine production in the anti-inflammatory direction to prevent excessive tissue damage. And this is very valuable since it is excessive reaction to infection that lead to the dangerous cytokine storms that will damage the tissue, like the linings of the lungs, and cause a lot of inflammation. And it turns out that as people age, they're more likely to have the vitamin D regulation become overactive. And so older people are more likely to die from infectious diseases than younger people are. I published a paper in 2009, sort of analysis of the effect of solar UVB and vitamin D on case fatality rates from the 1918-19 influenza pandemic in the United States. So what happened was the Public Health Service in the United States went to 13 communities and had people go door to door asking residents, did somebody in this household die during the pandemic? Did they get the influenza and did they die? And so that's a case fatality rate they were studying. And what I found was that the deaths were less in the South and in the Southwest, higher in the North and Northeast. And so that meant that if I looked at the July UVB dose, there was about a 28% reduction. Well, it was stronger reduction for that, and the rates of pneumonia as a complication were also very strong. Similar results found for wintertime UVB. The hypothesis was that UVB, through production of vitamin D, reduces the severity of influenza as well as greatly reducing the risk of pneumonia, the main cause of influenza. So they could kill a pneumonia virus as well as bacteria as well. So I published a paper in Nutrients in 2020 in April. Now, I published in Nutrients because Nutrients publishes very rapidly. It does have peer review, and it's very vitamin D friendly. So had I tried to publish this in a mainstream journal, it might have taken 6 months to a year and would have not been the first paper published pointing out that vitamin D could have played a major role in protecting people during the pandemic. So the evidence I use includes that the outbreak occurred in winter, a time when 25-hydroxyvitamin D concentration is lowest, that the number of cases in the southern hemisphere near the end of summer is low, that vitamin D deficiency has been found to contribute to acute respiratory distress syndrome, and that case fatality rates increase with age and chronic disease morbidity, both of which are associated with lower 25-hydroxyvitamin concentrations. So, as I mentioned, this did stimulate other research analysis, but because Big Pharma sees vitamin D as a threat to income and profit and to the vaccines that we're developing and the expensive drugs that we're developing for preventing and treating COVID, there was a media blackout on vitamin D. The medical system had doctors and hospitals not use vitamin D and other repurposed drugs, and the benefit was largely not available. However, if you come to 2024, a meta-analysis, in other words, a summary of all the studies on vitamin D supplementation using 16 publications, it found that vitamin D supplementation has a protective effect against the incidence of COVID-19, reducing it by 60 to 68%, 60% in general, and 68% regarding ICU admission. So this meta-analysis suggests a definitive and significant association between the protective role of vitamin D and COVID incidence and ICU admission. Turning now to another infectious disease, periodontal disease or periodontitis is mainly caused by infection and inflammation of the tissue around teeth. often causing shrinkage of the gums and loosening of the teeth. Vitamin D deficiency is a major cause of periodontal disease, as is poor diet. Vitamin D reduces risk through induction of human cathelicidin that kills the damaging bacteria, thereby reducing inflammation and by suppressing matrix metalloproteinases or extracellular enzymes known to increase gum destruction. It's been shown that periodontal disease is associated with 57 types of disease. I'm not going to even identify the disease. Well, it turns out diabetes is one of the diseases that has the highest correlation with periodontal disease. And it's been shown that there's a two-way link between periodontal disease and diabetes. If you get diabetes, you're likely to get periodontal disease and vice versa. The periodontists have made the claim that since periodontal disease is associated with all the diseases, and since it occurs often before the other types of disease, treating periodontal disease by cleaning the plaque off the teeth will help reduce the risk of systemic and other infectious diseases. Well, I disagree. They have not really shown through clinical trials that treating periodontal disease by their means really has long-term effects on systemic diseases. So we published this paper in Nutrients, and we're trying to convince periodontists and doctors and dentists and even medical doctors that they should treat this like the canary in the coal mine and tell people that, hey, you should be changing your diet, go to a healthy diet, go to which may be more of a plant-based diet, and cut out red meat and processed meat and so on, and processed foods and sugar and so on. And you should take vitamin D. Unfortunately, our medical system is set up to treat disease through pharmaceutical drugs and surgery and so on. And doctors, like I say, do not believe in vitamin D. They consider it the enemy. And so far, they have not accepted our proposal to make changes in their dealing with their patients. Now, pregnancy outcomes is another area, another type of health outcome that's where vitamin D is very, very important. Pregnancy represents a dynamic period with physical and physiological changes in both the mother and her developing fetus. The dramatic 2- to 3-fold increase in the active hormone 125 or dihydroxyvitamin D or calcitriol concentrations early in the weeks of pregnancy, despite minimal increased calcium demands during that time of gestation, and which are sustained throughout pregnancy in both the mother and fetus, suggests an immunomodulatory role in preventing fetal rejection by the mother. This is by Hollis and Wagner in 2017. And they are the primary, the best researchers on the effect of vitamin D during pregnancy. Another effect showing that vitamin D is very important for pregnancy is that women in all ethnic groups have lighter skin pigmentation, likely due to vitamin D requirements during pregnancy and lactation than men, and men are more likely to spend time in the sun. This is by Nina Jablonski and George Chaplin, who have spent years going around the world measuring skin pigmentation. This is the implications. One implication is that there's the Barker hypothesis, first described by David Barker in 1968, noting a connection between neonatal growth restriction and small for gestational age status with the risk of heart disease later in adult life, and has become mainstream thinking. Addressing the original question of this review, does vitamin D quality as a substance that supports and upholds the Barker hypothesis? The clear answer is yes. Through its effect on genetic processes, vitamin D deficiency during pregnancy affects both mother and fetus, and at least in the case of asthma, phenotypic expression in the infant or child. Now, Bruce Hollis took part in a study in Iran where they had a stratified randomized field trial. They had women in 2 hospitals, and they gave vitamin D supplementation in one of those hospitals. So 900 women in only 1 hospital were only observed, not supplemented with vitamin D. The mean 25-hydroxyvitamin D was 11 nanograms per milliliter throughout the study. Now, in the US and Europe, it's no longer possible to conduct a study where the control group is not given at least a minimum amount of vitamin D. So this will not be possible in the United States. Now, the 900 women in the screening site were supplemented with individual doses to raise their mean 25-hydroxy from 11 nanograms to 21 nanograms per milliliter. This is open access, published in 2018. And the results may be a little hard to read, but for preeclampsia, gestational diabetes, and preterm delivery, those who were in the severe deficiency had by about a 50% reduced risk of preeclampsia and a preterm, maybe a 70% reduction in preterm gestational diabetes and preterm delivery. Those who are in the moderate deficiency had non-significant reductions in several of these outcomes. So this confirms that vitamin D supplementation is very important in pregnancy. Here's a study conducted at the Medical University of South Carolina, the University of Hollis and Wagner. And they included 1,000 sequential pregnant women at their obstetrics center on their first prenatal visit. They measured 25-hydroxyvitamin and gave them free supplements of 5,000 IU and counseled how much to take to achieve 40 nanograms per milliliter. Of course, not all the women did. And what they showed was that those who achieved less than 20 nanograms per milliliter at delivery had about a 37 to 38 week delivery, whereas those who achieve greater than 40 nanograms per milliliter had more like a 38 to 39 week delivery. And they found that the results were independent of race, and they found a 60% lower preterm risk for the patients with greater than 40 versus less than 20 nanograms per milliliter. In terms of diabetes, there was a study at Tufts University called a D2D. It was preterm, I'm sorry, got to change diseases. It was pre-diabetes. Did they go to diabetes with vitamin D supplementation? Now, in this case, they actually supplemented with 4,000 IU per day. And in the original analysis published in 2019, they found that comparing those who were treated with those who were not treated, there was no significant effect on reducing conversion to new onset type 2 diabetes. However, they did a reanalysis looking at the intra-trial vitamin D exposure and serum 25-hydroxy concentration. And they found that for those with, they could look at different levels from less than 20 to 30, 40 to 50, and greater than 50 nanograms per milliliter compared with less than those 20 to 30 nanograms per milliliter. And what they found was that for each increase of 10 nanograms per milliliter in the intra-trial 25-hydroxyvitamin D level, that was associated with a 25% reduction in those assigned to vitamin D, and a non-significant 10% among those assigned to placebo. And they found that those who achieved greater than 50 nanograms per milliliter had a 71% reduction compared to those who maintain a level of 20 to 30 nanograms per milliliter. So indeed, high-dose vitamin D supplementation and achieving a high level does reduce the risk of conversion from prediabetes to diabetes. The authors say this is only for prediabetes. I think it might apply to diabetes in general. On the other hand, vitamin D is not the only factor that affects diabetes. I've done other work, and I've looked at other work from Harvard showing that it's really important to have a diet that reduces inflammation and reduces insulin resistance. And such a diet would be low in red meat, low in processed meat, low in the ultra-processed foods. It would be high in green leafy vegetables. Coffee is okay. Have colorful fruits and vegetables. It would have a lot of whole grains, some fish, some legumes, et cetera, et cetera. So one really has to work with both vitamin D and diet to reduce the risk of diabetes. There are mechanisms identified whereby vitamin D reduces insulin resistance. So vitamin D exhibits indirect antioxidant properties and participates in the maintenance of normal resting reactive oxygen species level. Appealingly, vitamin D reduces inflammation and regulates calcium level in many cell types. Therefore, the beneficial effects of vitamin D include diminished insulin resistance, which is observed as an improvement of glucose and lipid metabolism in insulin-sensitive tissues. Turning now to cardiovascular disease, there are four main types: coronary artery disease, or coronary heart disease, cerebral vascular disease, peripheral arterial artery disease, and aortic atherosclerosis. Not going to the other details. This is a study from Utah looking at the survival curves of cardiovascular disease patients over a 6 or 7 year period. Those with greater than 30 nanograms per milliliter had an 84% survival. Those with less than 15 nanograms had a 70% survival. Now, this is actually all-cause mortality rate. It's not cardiovascular disease mortality rate, but it is patients who had cardiovascular disease. So the evidence that vitamin D reduces risk of cardiovascular disease is not as strong as, say, for cancer, as I'll get to. We do have another observation on stroke. This is a 45, a cohort of Black and white adults greater than 45 years old. It was a case cohort study, 610 participants. And what they found that those with greater than lower concentrations were associated with higher risk of stroke, with greater than 30 nanograms per milliliter as a reference. Those with 20 to 30 nanograms per milliliter had a non-significant 33% increased risk. Those with less than 20 nanograms per milliliter had a significant 85% increased risk. Importantly, there are no statistically significant differences in the association of lower 25-hydroxy vitamin D with higher risk of stroke in black versus white participants in the fully adjusted models. Now, here is a Mendelian randomization analysis finding an L-shaped association of serum 25-hydroxy D and cardiovascular disease. This was stratified, so they looked at the genetic score for 25-hydroxy out genetic score out to a difference of 12 nanomoles per liter. And they found that the effect was very strong below 25 nanomoles per liter or 10 nanograms per milliliter. They could show about that 6% of the cardiovascular disease risk can be prevented by increasing 25-hydroxy D. But you take studies like the VITAL study, which enrolled people starting with a mean value of 30 nanograms per milliliter, they're not going to see an effect that occurs below 10 nanograms per milliliter. So that's, I think, part of the reason we don't have good evidence that supplementing with vitamin D reduces the risk of cardiovascular disease. Turning now to autoimmune diseases, these are diseases where the body's immune system attacks the body. Common autoimmune diseases include celiac disease, inflammatory bowel disease such as Crohn's disease, ulcerative colitis, lupus, multiple sclerosis, rheumatoid arthritis, type 1 diabetes mellitus, and thyroiditis. Can vitamin D reduce the risk of autoimmune diseases? Well, type 1 diabetes mellitus rates were very low in Finland for infants given 2,000 IU per day in the first year of life, as reported in 2001. In a later paper, it was reported that as the vitamin D dose was lowered, the rate of type 1 diabetes in infancy increased significantly in Finland. Now, the VITAL study did look at autoimmune disease incidence, and they found that for vitamin D arm, 123 participants in the treatment arm and 155 in the placebo arm had confirmed autoimmune disease. This is a significant 22% reduction. This was published 3 years after the main publication. And it turns out rheumatoid arthritis is one of the stronger diseases found to have an effect. I'll skip this one. Now, in terms of racial disparities, I published a paper with Bruce Ames and Walter Willett in 2021. We summarized the evidence that the health disparity is partly due to insufficient vitamin D production caused by melanin in the skin blocking the UVB solar radiation necessary for synthesis. The vitamin D insufficiency is exacerbated at high latitudes because of the combination of dark skin color with lower UVB radiation levels. And the health of individuals in dark skin can be markedly improved by correcting deficiency and achieving an optimal vitamin D status, as can be obtained by vitamin D supplementation and or fortification. So this is the data on comparing mean 25-hydroxy D concentrations by race in the US. The blacks are in dark, Hispanics in gray, and whites in white. So you see for serious deficiency, blacks, 17% have serious deficiency compared to 1% of whites. 58% of blacks compared to 19% have deficiency. That's 25 to 50 nanomoles per liter, or essentially below 20 nanograms per milliliter. 21% have insufficiency, that's between 20 and 30 nanograms per milliliter, and whereas only 4% have greater than 30 nanograms per milliliter compared to 35% for whites. So we look at percentage increase in cause-specific mortality. for Blacks compared to white Americans aged 50 to 65 years. And for all cause, you see about a 45% increase, heart disease, a 70% increase, cancer, 140% increase, cerebral vascular disease, around 35%, and diabetes, 100%. So not saying that all the difference is due to vitamin D, but it contributes a significant amount. Now, unfortunately, since doctors do not would rather treat people with disease than prevent disease in the United States, not even the African American Medical Association with 50,000 physicians has accepted vitamin D as beneficial for their patients. So we said that the diseases with reasonable evidence that vitamin D supplementation reduces disease include all-cause mortality rate, adverse pregnancy and birth outcomes, many types of cancer, type 1, type 2 diabetes mellitus, Alzheimer's disease and dementia, multiple sclerosis, acute respiratory tract infections, COVID-19, asthma exacerbations, rickets, and osteomalacia, which is a form of bone disease. Now getting to reviews. Here's a review published as a result of a vitamin D conference in Warsaw. Paweł Pudowski convenes experts from Europe and the United States every 2 years since 2012 to review the effects of vitamin D and the benefits. So this is one of the more recent reviews, and says, according to data from RCTs, a daily dose of 2,000 IU per day is sufficient to maintain serum 25-hydroxyvitamin D concentration of 50 nanomoles per liter, 20 nanograms per liter, and above 75 nanomoles per liter in greater than 99 and 90% of the general adult population, respectively. According to large vitamin D RCTs, there are no significant safety concerns in supplementation for such a dose for several years, even in individuals with an already sufficient vitamin D status at baseline. And recall, there are mechanisms whereby vitamin D can be normalized, reduced, or not gotten too high if people also have sun exposure. So the result is a daily vitamin D supplementation with 2,000 IU per day may be considered a simple, effective, and safe dosage to prevent and treat vitamin D deficiency in the adult population. Now, on the other hand, I published a narrative review of the evidence for variations in serum 25-hydroxyvitamin D concentration thresholds for optimal health. Now, this is based on observational studies rather than RCTs. The general finding is that optimal 25-hydroxyvitamin D concentration support health and well-being are above 30 nanograms per milliliter for cardiovascular disease and all-cause mortality rate, whereas the thresholds for other disease outcomes appear to range up to 40 or 50 nanograms per milliliter. And the most effective way to achieve these concentrations is through vitamin D supplementation. This is a table from that study, perhaps hard to read, but it shows Alzheimer's disease greater than 25 nanograms per milliliter, breast cancer greater than 60 nanograms per milliliter, colorectal cancer 30 to 40 nanograms per milliliter, cardiovascular disease greater than 30 nanograms per milliliter, myocardial infarction or heart attack greater than 30 nanograms, SARS CoV-2 infection. This is an observational study showing 50 nanograms per milliliter is recommended. COVID-19 mortality greater than 60 nanograms, although most of the effect is for people who had baseline 25-hydroxy vitamin D of less than 12 nanograms per milliliter. Diabetes mellitus type 2, greater than 50 nanograms per milliliter. Gene expression, greater than 40 nanograms per milliliter. A study on hypertension showing that people who are hypertensive and were already taking vitamin D and had a mean 25-hydroxy vitamin D of 30 nanograms per milliliter reduced their prevalence of hypertension and blood pressure by going to above 40 nanograms per milliliter. And finally, preterm delivery showing that greater than 40 nanograms per milliliter is beneficial. Now, to function properly, vitamin D needs magnesium, calcium, omega-3, boron, zinc, and vitamin A and K2. Magnesium is one of the most important cofactors, and to put it simply, it is necessary for vitamin D to bind to its transport proteins and be delivered to the tissues. Magnesium is synergetic to vitamin D3 and increases benefits up to 30%. While vitamin K2 is another important component needed to work with vitamin D in the metabolism of calcium, so that calcium is deposited in the teeth and bones rather than in the blood vessels. K2 vitamin is also essential for bone mineralization. So vitamin K2 can be obtained as a supplement. It can also be obtained in nattokinase, which is a fermented soy product. It's more important for people taking high dose vitamin D to help make sure the calcium is deposited in teeth and not in the blood vessels. So one asks, are there adverse effects of vitamin D supplementation? The most important adverse effect of supplementing with excessive high-dose vitamin D doses is hypercalcemia. The symptoms include digestive symptoms such as nausea or vomiting, poor appetite or constipation, increased thirst or more frequent urination, muscle weaknesses or twitches, changes in how your brain works, such as feeling tired or fatigued or confused, bone pain, and fragile bones that break more easily. This really only happens primarily if you're taking 50,000 IU per day based on not understanding that's not a daily dose, or taking even up to 1 million IU per day due to manufacturing and labeling errors. So here's the results of finding somebody who had been taking 1 million IU per day and achieved greater than 900 nanograms per milliliter, to the point where he had bloody feet, could not think straight, had to cancel his lecture tours. So it took about a year to normalize the 25-hydroxyvitamin D levels. However, once the 25-hydroxyvitamin D levels decreased below 400 nanograms per milliliter, both patients became normal calcemic and asymptomatic without long-term sequelae. So they didn't die, and they survived. Now, there are non-vitamin D beneficial effects from sun exposure, and I work mainly with the vitamin D effects. In a recent review by Slominski et al., the UV ray effects induce electrical, chemical, and biological signals to be sent to the brain, endocrine and immune systems, as well as other central organs, which can regulate body homeostasis. Such modulatory effects will be dependent on UVA and UVB wavelengths. This leads to immunosuppression, the activation of brain and endocrine coordinating centers, and the modulation of different organ functions. Some of the non-vitamin D effects of sun exposure include that it increases serum nitric oxide concentrations, which reduces risk of cardiovascular disease and infections. Richard Weller in the UK has published a number of papers on this topic, and the Nobel Prize for nitric oxide was given in the late 1990s, but it was only in the after around 2010, 2015 that it was realized that solar UV through production can liberate nitric oxide from the nitrate stores under the skin. Blue light helps reset the circadian rhythm so that morning or evening sun exposure helps with this. Sunlight reduces risk of seasonal affective disorder that happens in spring and fall. It reduces endorphins and opioids so that it feels that sun exposure gives pleasure. And sun exposure increases steroids, cortisol, corticosteroid, and pregnenolone. So for further reference, I do most of my initial searching at Google Scholar, scholar.google.com. Google Scholar has connection, they link to nearly all of the papers, scientific papers published, and it's easy to search. You just put in keywords like vitamin D and a topic like cancer, and then Google Scholar will give you the results. They'll try to give you the most important ones first, and you see that they're important because of the number of citations they have. And then Google Scholar will also link to the papers and show you where you can get the paper, a free downloadable paper. It can also give them by years, so you can say, well, let's give the range from 2022 to the present. And so it's a really good search engine. Now, PubMed is okay, but it only it has the journals it thinks are good, like it does not link to journals on fluoride because the CDC is promoting fluoridation of water, not trying to eliminate fluoridation. So now PubMed, I also use. I go there every day. It might have the documents faster than Google Scholar does. Next, there's vitamindwiki.com. This is a website that Henry Lahore, a retired Boeing aircraft engineer, has been maintaining for 5 to 10 years, 10 years at least. He spends every day sucking in the latest information on vitamin D, making it accessible and searchable for the general public. He's in Washington State, and it's just a very, very good website. Then there's grassrootshealth.net. This was started by Carol Baggerly in around 2008. She is a breast cancer treatment survivor. She went through all the treatment for breast cancer, and being a good researcher. She was a businesswoman who did a lot of research in her business before she went to companies to tell what she could be doing for them. When her general practitioner told her that she needed to start supplementing with vitamin D, she researched and found that Cedric Garland, who was in San Diego or La Jolla, had published many papers on vitamin D and breast cancer. And she contacted him and found out, and she was just terribly disappointed that the medical system had not alerted her to vitamin D earlier in her life, which might have prevented her breast cancer. So she made it her mission to form this organization to help promote vitamin D to the general public. She's now retired from active role in that organization, but it's well run by the staff that has maintained it. And if you want to have your vitamin D level measured, they will, for about $70, they will send you vitamin D test kit. Finding my own. If you want to get more information from me, my website is WBGrant. My email address is wgrant@infyonline.net. Okay.
All right. Thank you. So you're done with your presentation, Dr. Grant?
Yes, I'm done. Thank you.
All right, great. Thank you. So now we will begin the Q&A portion of the presentation. And okay, so before we do, besides your email, do you have a website or can people follow you online?
Yeah, yes. I'm on Twitter at my handle is WBGrant2. And I actively promote vitamin D there. I also have a website, sunarc.org, sunarc.org. So you can find me that way. And my book is now available for free through my Twitter account, my X account.
Okay. So you can get a digital copy of your book. Is that what you're saying?
That's correct. That's correct.
Okay. And then people can also get the book through our website, therealtruthabout.health.com, and go to bookstore, and you'll find Embrace the Sun by Dr. Grant.
Okay. It's no longer available for sale as a hard copy. That's why it's now available as a
okay.
copy.
Thank you for that clarification. So now let's go ahead and begin with the Q&A. If the audience would like to ask questions, you can simply raise your hand in Zoom, and we'll pick on you when we get the opportunity to. So how do you address studies that suggest a strong link between sun exposure and increased risk of melanoma?
Well, there is a link. What we try to tell people is that you have to have sensible sun exposure. Try to avoid burning. Try to if you're going to be working in this outdoors, you might want to raise your vitamin D levels through supplementation, because that will help. If you get above 40 or 50 nanograms per milliliter, that will help protect you against burning and cancer. And it turns out that those who develop melanoma have the best outcome if they have high vitamin D levels. It turns out that since melanoma is a cancer, vitamin D reduces the progression and mortality just like it does for any type of cancer.
So should everyone be supplementing with vitamin D?
I would say yes, because most people think that if they're in the sun, if they work outdoors or something, they might have adequate vitamin D levels. But if they cover up, women, for example, use a lot of makeup, and makeup now has vitamin D skin protection, UV block. If they're out in winter, they're not making vitamin D. If they're out when the sun is not too high, they're not making vitamin D. If they wear too many clothes, they're not making enough vitamin D. So it's really, and the benefits of vitamin D do accrue even up above 30, 40, 50 nanograms per milliliter. And I'd say going up to around 50 nanograms per milliliter, you're safe and having a better health outcome.
So sunscreen, with what you were talking about in your presentation, so sunscreen blocks UVB, which gets converted into vitamin D, and it doesn't block UVA, which causes cancer, right? Is that a correct understanding?
Well, it doesn't block UVA as well as UVB, and the UVA is what causes melanoma and basal cell carcinoma. Squamous cell carcinoma, which is the most serious skin cancer other than melanoma, is caused by UVB, as are cataracts. So if you're going to be out in the sunlight, you want to wear glasses to protect against UVB hitting your eyes. But now it's only UVB reduces the risk of internal cancers. UVA doesn't have any effect on internal cancers.
And what about skin burning? Is that UVA versus UVB? Does it matter?
That's both UVA and UVB.
Okay.
But it's probably more, it's more the UVB and the shortwave UVA. So the skin, most of the transparent skin sunblocks go into the the shortwave UVA, but they still let the long wave UVA transmit and the visible.
So when should someone use sunscreen? If they're going out for a little bit, obviously a little bit is good for you. When do you think?
You might want to put it on after you've been in the sun a little while, so you can get the benefits of UVB.
And are there sunscreens that are better than others? Some of our guests are talking about zinc as opposed to these chemical-based sunscreens. Any thoughts on that?
Well, dermatologists, there's a picture of a dermatologist in Australia putting on zinc oxide. His face is painted entirely white. The vitamin D levels of dermatologists in Australia are very low compared to the average average Australian. I mean, it's zinc oxide is going to block all the UVA and UVB, but I don't think that's necessary. I don't know which of the sunblocks are better for UVA. And if you want to make sure you have UVA protection, maybe the zinc oxide is a better way to go. Or just wear clothing.
Okay, so to cover yourself up. And is vitamin D more important at some ages than at other ages?
Yes, it's very important in the first year of life. It's very important during pregnancy and reproductive life. It gets more important as time goes on. If you look at all the diseases like cancer and the severity of infectious diseases, they start increasing above the age of 50 or 60. And if you look at COVID, the people most likely to die were over the age of 70 or 80. So you probably want to make sure you're supplementing as you age.
And so what would be the appropriate dose if you weren't getting your blood checked right away and you just wanted to have a safe dose, given that almost everybody is deficient in D, given our current lifestyles?
Okay. So the Institute of Medicine in 2011 recommended 600 IU per day for people up to the age of 70 and 800 IU per day for people over the age of 70. But in 2011, they were interested in two things. One was they only had results for bone health. They also had observational studies that showed that people in observational studies who enrolled with higher 25-hydroxyvitamin D levels had higher mortality rates than people with intermediate levels. And they didn't understand that what happened before that period was that as people came in to see doctors in the early 2000s, if they were found to have osteomalacia or osteoporosis, doctors said, now start supplementing with vitamin D. So they lived their entire life up to 60 or 70 or so without supplementing with vitamin D. And they probably developed diseases related to vitamin D deficiency. And very quickly in the observational study, they died, but it was probably related to vitamin D deficiency earlier. They were also at that time concerned about the VITAL study, and they were going to recommend that anybody in the VITAL study could take as much vitamin D as they were recommending in this 2011 report. So they figured if they recommended 2,000 IU per day, that would destroy the VITAL study. Well, also in 2011, Michael Hollick convened a study group for the Endocrine Society in the United States, and they recommended getting above 30 nanograms per milliliter, whereas the Institute of Medicine said only above 20 nanograms per milliliter. So what we've shown now is that everybody should be supplementing with at least 2,000 IU per day. I've been supplementing with 5,000 IU per day. which got my level up to around 60 or 70 nanograms per milliliter. But I'm a small person weighing about 130 pounds. Obese people need more than thin people. So 4,000 IU per day for obese people would be more what we'd recommend than 2,000. If you're African American, 4,000 would be more recommended. Hispanic, between 2,000 and 4,000. So it depends on several factors, but those are some of the considerations.
And there's also discussions in this world with liquid versus the sublingual or the tablets. What is the best way to supplement with vitamin D?
Okay. It turns out it doesn't matter whether you take vitamin D with the meal, with fat, without. Orange juice is fortified with vitamin D. Bread is fortified. Milk is fortified. There's a powder version. There's also the version where vitamin D is in a lipid of fat of some sort. It doesn't really matter. The important thing is that it's a reputable manufacturer, and that the vitamin D on the dose is what's on the label is what's in the dose.
Okay, and
taking vitamin D in the middle of the day might be a little bit more better than taking it in the morning and evening.
Why?
Evening it might affect with sleep as you're trying to metabolize the vitamin D.
Okay,
and if you're measuring your vitamin D level, you probably want to measure it during the day, middle of the day as well.
Okay, so I think one of our speakers had said something about vitamin D being fat soluble. Is that not true then?
Yes, excess vitamin D can be stored in the fat tissues, but it is, and if you start reducing weight and dissolving the fat, you're going to raise your vitamin D levels, because it's going to then go back to liver and convert to 25-hydroxyvitamin D. But I mean, it's not like vitamin C that's in and out. It's water soluble, it's in and out very quickly.
Okay. And what are the most convincing studies that support your stance on the benefits of vitamin D, either through sun exposure or supplementation?
Well, I published a paper in 2002 with Alberto Munoz, is an open access paper at Nutrients, on a historical overview of vitamin D in cancer and the mechanisms. And we go through all of the epidemiological studies, the clinical trials, the observational studies, the mechanism, and we show that there's a general agreement, if you look at all the studies, that sun exposure and vitamin D reduce the risk of cancer incidence and mortality rates. So that's cancer, infectious diseases. So I published the paper in 2020, hypothesizing that vitamin D would reduce the risk of COVID based on mechanisms and effects for influenza and pneumonia. And the latest clinical trials and observational studies show that indeed vitamin D does reduce the risk of COVID incidence and mortality.
Great, thank you. So with that concludes our time. So thank you so much for that great presentation, extremely informative. If we could unmute the audience so they can say thanks.
Thank you so much, Dr.
Appreciate it.